All right. So, oops, let me just mute this. As promised, here's a quick video on some of the concepts from our vision lecture 2 weeks ago, and as you remember, this comes from a special request from one of your classmates who shall remain anonymous, but I guess some of us already know who that is. But I do appreciate it when students let me know there is a problem, and I don't mind taking half an hour of my time to go over some of these things, because of course there are a lot of new terms and a lot of new concepts here. So the student has gone ahead and sent me 4 kind of key areas that that would be good to have reviewed. And how I kind of decide to go about this is to make sure any slides that I do present in this little recording actually don't become anything new for you. So these are actually slides taken from the lecture. So there are no new visuals. And so if anything, if you're using this video to kind of review some of these concepts, you can kind of go back to the same slide from that week and see if there's anything else you needed to add on. All right. Okay. So we'll go and do this in order. Okay. So let's start with the first one. And the first one's quite in fact, quite straightforward. And the question, I literally just got points. So I'm just going to kind of, extrapolate as I think in terms of where the problems might be. The first one that I got was retinofugal projection. And this just simply refers to tracks immediately leaving the eye. So if we remember sort of where we were with the important part of the eye that sort of starts the whole cascade. I don't actually remember. Oh, it's not F12, it's F11. We remember that light enters from the lens, and eventually what we really care about is this retinal tissue that is at the back of each eye. And if we zoom in a little bit, of course, we learn that we've got a bunch of layers. The layer that we kind of care about is the backmost layer. Okay, the backmost layer that contains your photoreceptors, the cones and the rods. So of course, there's this whole thing with phototransduction. Eventually, this light energy becomes electrical energy and must get carried off towards your cortex. But of course, before it hits cortex, it makes a few pit stops. And so retinofugal projection is referring to really 3 chunks, 3 different segments of the initial departing white matter tract. The first chunk is the optic nerve, and this really just refers to this segment here. So it's from the retina to the chiasm. That's the optic nerve. And the second chunk that we do consider as part of the retinofugal projection is the chiasm itself. And then the third chunk that is kind of hidden by my head right now. Let's see if I can actually get to it. There you go. You see that? That's the optic tract. And the optic tract is specifically between the chiasm and then it's kind of hard to see here because it's inside here. This is just your brainstem here, but it's between the chiasm and the LGN in your thalamus. All right. That is the optic tract. So in general, when we're talking about the retinofugal projection, it's these 3 components. So that's pretty easy. And of course, what I made a point of saying at that time in the lecture is that what it doesn't include is, for example, the later tracks from the LGN to V1. So those we call the optic radiations. So it's important here because terminology does matter. Okay, so it's not anything leaving the eye can be called an optic tract. The optic tract specifically refers to this segment between the chiasm and the thalamus, and the optic nerve is this specific segment here. Okay, so they can't be used interchangeably. So in terms of retinofugal projection, then this is what we're talking about, just these immediate 3 chunks leaving the leaving the retina. And I suspect that's not where the problem is, though, right? I suspect the problem is more about this. If we really look at, the optic nerve segment here, and also then the optic tract segment, we talked about how, in fact, there is kind of this coarse division in terms of, if we think about the nerve, let's talk about the nerve first, as kind of being split into two halves, a nasal half and a temporal half, right? So the nasal half being closer to your midline or your nose, and then the temporal half on the outside, that we find each kind of segment, each half carries or is responsible for carrying data from different parts of the visual field, right? I suspect this is sort of where the problems kind of began with the retinofugal projection. And so remember, actually, I don't know if I still have that. Oh, I do have this image, right? This, the way to think about it, if we take the nerve and we divide it into the two halves, is that we have the two nasal halves here, right? Carrying data from a slightly different portion than the two temporal halves. Okay, so if you kind of want to follow the projection here, and let's just talk about the right eye in this case. If we talk about the back of the retina, and then we have a nasal portion, nasal representation that is being kind of carried off kind of on this end, you see that the nasal portion is actually carrying data from the rightmost, so ipsi periphery, and also a chunk of this kind of central vision area. Whereas the temporal retina of this right eye doesn't actually carry any data about the ipsi peripheral field, but rather encodes, if we will, or carries or represents data from, the central central field of view, but also some of the contralateral field of view as well. So similarly, if we look at the left eye, it's the same sort of cartoon, right? So we have the nasal half of this eye carrying data from the ipsilateral visual field, as well as some of the central part of the visual field, and then we have the temporal chunk carrying data from central vision, and also a chunk of the contralateral visual field. And I think when you're studying, this is sort of the image to kind of digest. I hate to use this term memorize, but unfortunately you will have to memorize this, because then understanding this, you don't really need to study kind of this slide, because so long as you understand what chunk each of these fibers are of the visual field are carrying, then you can kind of draw it out and deduce, hey, if I lesion here, then I'll lose this chunk, right? So remember, of course, that at some point, and this is represented here, we have to consider what's happening at the chiasm. And so when you're kind of studying the slide, remember, the tricky part is that the nasal side of each nerve actually eventually crosses over to the contralateral brain. So you can see kind of this blue line carrying over from the chiasm over to the other side. What this means is, by the time we're talking about what's this chunk again, right? This chunk is, okay, your optic tract, right? This was the nerve, this was the tract, and then this is showing you the optic radiations. But the point is, after the chiasm, we already have a situation where the kind of left side, okay, and just follow this trace. The left side data is actually what? The left side data is in fact representing here. Okay, so this includes the right, so the contralateral visual field, including the contralateral periphery, and of course the original ipsilateral temporal data, which has stayed on the same side, right? So it's really carrying data from from the central and contralateral visual field by the time we have crossed over the chiasm. So similarly, again, so on the right eye here, okay, so remember what's happening, okay, so of course our temporal data stays ipsilateral, okay, regardless of what's happening at the chiasm, but then the other eye's data, okay, is crossing over. The other eye's nasal data is crossing over. So what do we have again? You can just follow kind of the shaded region, right? What was the nasal region or nasal fiber carrying data of? Again, it was carrying data of this ipsilateral peripheral and central hemifield, right? So once it crosses over, again, what do we have? We have all the red stuff, okay? Once it crosses over past the chiasm, then we have a representation of the full contralateral hemifield. So I mean, in terms of making predictions here on this next slide, which is my gut is where we start losing some of you in the lecture. This is very easy to predict once you are very, very fluent with sort of what's happening with the nasal and temporal, and then remembering that both nasal fibers cross over at the chiasm. All right, so this is going to be the slide and image really of study. All right, okay. So now we're going to get to the second concept, this topic of retinopathy. This one's actually not that bad. What this simply means, and I think this is the image to kind of pay attention to. What this simply means is if we sort of break down your entire visual field into a grid, right? And I'm just going to coarsely, let's not bother breaking it down into horizontal grid. Let's just split it up here from left to right, you know, number 1 chunk, number 2 chunk, all the way to number 9 chunk. So this is the world that you're looking at. The concept of retinopathy simply means that there is a physiological correspondence between, if I'm fixating here at 5 and I give you a stimulus at 1, okay, that will always invariably result in stimulation at this point on the retina, okay? And this point number 1 on the retina will also invariably elicit activity in some corresponding location on the LGN, and this point of stimulation will then translate to some specific point of stimulation at V1 as well. So there's this kind of physical correspondence between the visual space, the part of the visual space that is being stimulated, to the portion of the retina that's stimulated, to the specific portion of the LGN that's stimulated, to the specific portion of V1 that is stimulated. And that's sort of the thing to kind of note here. There's this concept of topography. Okay, there's this spatial preservation. And it's very orderly. So if then I'm fixating at 5, but then, you know, position number 2 is stimulated, okay, that will always elicit kind of a stimulation just neighboring to position 1, etc. The whole cascade follows. I think I remember now, like at break in that lecture, there was a student that then came up and said, well, but doesn't the kind of location of the visual field that the stimulus falls on depend on where you're fixating? Exactly. It absolutely depends on where you're fixating, right? So this is why, do you remember I was showing you those rotating wedges when we go and try to map retinopathy in the MRI, for example. So for the assumption is if I'm fixating in the center, okay, let's just bring up my thing again. The thing I was showing you was, I think, like, and typically in the lab we have, a fixation. And then what we do is we tell the subject, please, for the love of God, if you can't do our task, please fixate. And then what we're going to do is we're going to just keep rotating our position of stimulation. So the wedge just keeps going around and around. Now, where trying to map retinopathy fails is when the subject starts moving their gaze. right? So then in this case, when I move my gaze, okay, then, the location of 1 is no longer going to be the proper location that I was looking at earlier, right? So yeah, absolutely. So this everything is, when we're talking about positions 1 to 9 here, okay, we're talking these positions assuming that, for example, you are fixating at number 5. All right. Okay, so hopefully that's clear. This magnification here is just something else entirely. It's another property of the visual system that I introduced here, saying, okay, well, the other thing is, not only do we have this orderly spatial representation physiologically, but we also have this kind of over-representation of central vision. So and this is kind of, do you remember I kind of cropped this out and turned it around to show you that the kind of the location, the central location was kind of exaggeratedly represented in V1. So that's what this cortical magnification refers to here. All right. So basically, if I'm fixating at 5, then it will be this kind of 5 location that will have the most cortex dedicated to it. And functionally, this is also why we find that certainly when I want to see something clearly and in more detail, I need to gaze to it, right? I need to foveate to it. But of course, what this also means that as I'm dropping off from 5, if this is where, again, I'm foveating, as I'm going further and further to the periphery, I have less and less cortex dedicated to it. Okay, the visual system's funky in that manner. It knows sort of where to prioritize. And so, that's again, this is sort of distinct from the concept of this spatial topography, this retinotopy, but it is an important concept to remember as well. All right, so moving along. I have to get rid of this drawing. Moving along. Oh yeah, this is the stuff I was putting up. Okay, ocular dominance. This is interesting because I think it's actually something. Sometimes I get questions in email and then I forget to share it with the class the following week because we just moved on to a different topic. But I do remember there's a question here about wait, hey, these images are sort of incongruent. Because, we have an image here that seemingly shows you ocular dominance existing across all 6 layers of cortex. And then suddenly, you kind of remember there was a textbook image that shows you, okay, there's actually ocular dominance in layer 4, which is this one here. And I think the question that week was, hey, those two are kind of incongruent, right? Does ocular dominance exist across all 6 layers, or is it just in layer 4? The answer is you have to be very, very careful with this. Ocularity, I think I use this concept of ocularity in terms of being purely responsive to one eye, right? That is indeed in layer 4. Okay, so pure ocular segregation, I think this is what I said in the lecture, exists in layer 4. So that means, yes, if we go and peel apart layer 4 and we just look at it perpendicularly, for example, we'll have the blue encoding, left eye, and then the kind of beige doing the right eye, and then we'll just keep alternating like that, right? And so then what happens is after layer 4, for example, as we rise to layer 3, I think you remember me saying this is where binocular neurons first emerge. And so what we mean by binocular neurons here is I will be responsive to both eyes stimulation, okay? Not just responsive to the right eye, for example. So layer 4 neurons would be purely ocular segregated. So these neurons will only respond to right stimulation, and these neurons will only respond to left stimulation. So what will happen as binocularity emerges in the outside layers, okay? We still do have ocular dominance in the sense that we still have a preference. If I'm now, let's just use this cube this time, okay? And we said layer 4 is pure ocular segregation. And layer 3, well, what happens? Okay, if I'm a neuron located right above this okay, so let me draw this. Okay, so let's say I've got one neuron here, and I know that I'm right, purely right segregated, okay? Because I'm in layer 4 also, and I'm in the right column, okay? Now let's say we pick a neuron that's directly above it in layer 3. These neurons will be binocular, so it will respond to both left and right eye input, but it will prefer right eye input, okay? Similarly, this neuron here, this one here, okay, will be binocular. It will fire a little bit to both right and left eye input, but it will prefer left eye input. So this is why ocular dominance columns are often shown like this across all 6 layers, because yes, ocular segregation, pure monocular neurons, layer 4, but then outside of it, you can kind of sort of visualize that we still have ocular preference. Okay, so this is why when we talk about ocular dominance, we generally say, it's ocular dominance column coming down the entire vertical patch of the 6 layers, and so hopefully that kind of clarifies that. Okay, so where are we now? Don't know why sometimes it doesn't change. Oh yeah, so this is what this slide was about here. So that's the point I was just making. So this is a pure monocular 4C neuron to the left eye, and directly above it, okay, these will be binocular neurons, but it will be, left preferring. Okay. All right. Oh yes, the last point here has to do with the pinwheel and cytochrome oxidase blobs. So we've talked about, so this wasn't a point here, but of course the first thing we talked about was orientation columns across cortex. And then the second thing we talked about a little bit there with the eye preference columns, right? So the ocular dominance columns. Of course, there are more properties at the visual cortex that we haven't covered. But one thing to also, if we conceptualize, actually, I don't think I have the cube here. I don't. But, one of the most popular ways of teaching kind of how to understand all the properties of the visual cortex is to conceptualize. Oh, I'm terrible at drawing. We all know that, though, if you've been at my other courses. To conceptualize a patch of cortex as this cube. Right. And we think, we've got like layers 1 to 6 here. Okay. But then we've also got, in layer 4, we've got running perpendicularly. Okay. The ocular dominance columns. And then we've also got, do you remember the orientation columns coming down vertically, right? There's also another property you can kind of visualize in this cube, and that's this. If we kind of take all our orientation columns, and in fact, I think a part of the confusion might be you think they're sort of like just all orderly in kind of like a a straight line strip. But if we're thinking about how the orientation columns are are organized from, like, let's say reviewing from the top surface of the cortex. Okay, what we find is that, let's say here, this is very similar. It's like an optical imaging method here. But if you just kind of loosely understand, without caring about the technique, that each color here is coding for an orientation preference. Okay, an orientation column, and it's kind of showing you here, red is this kind of horizontal orientation, and this light blue is this vertical orientation, et cetera, et cetera. And if we kind of look downwards and we're just looking at the surface of, layer 1, whatever, right? And we actually find that there's really this nice smooth transition from orientation to orientation, and it's not necessarily going kind of like a straight line, but you do kind of find that they do eventually form these pinwheel type patterns. When the original paper on this came out, I think it was like early 90s. In fact, I know it is early 90s. And this was kind of visualized in the monkeys. What they found was that, hey, if we zoom in a little at these patterns, these orientation patterns, we zoom in a little, we find that there will exist these kind of, they called them back then, these nodes or these columns of singularity. And sometimes they call them these columns of iso-orientation. I think for you, the easiest way to understand this is, okay, now I'm looking down from the surface and I'm just gonna zoom in a little bit. And what we find is that there are all of these smooth transitions, but they're always, at certain points, we can find these columns where exactly we have a radial representation of all possible orientations. So here I'm just zooming this out, and if we have a look, we're really just smoothly transitioning across all possible orientations, and in the middle we find this column, again, this column of singularity where we have exactly one representation of each orientation. Now, why this is relevant is because it's not just about, oh, hey, it's nice, all these orientation columns converge in this kind of radial way. We have this kind of center, stem point. That's not the point here. It's not just about, hey, it's cute, it's a pretty picture your view one is making. It's because coincidentally, right at these pinwheels, these columns of singularity, where we have just sort of like, one representation of each orientation, just this one iso zero orientation preferring kind of characteristic, is where we also find these kind of color preference or color responsive properties. And so these are the CO blobs. I think on the next slide, I think I might have. Yeah, so this is the one that I didn't show in the lecture, but so this indeed was from the early 90s, and you do find, so now what we're showing you is, okay, now I have a lot going on, right? Because I've got orientation columns that I'm looking off from the top, and then we're saying, okay, they make these funny spiral radial like shapes, and then in the middle of the singularity point, it turns out here we have color responsive columns, all right, these CO blobs. What I want to show you, so to help visualize, is of course down there in the rest of the cube, we've still got those alternating ocular dominance columns. And so what you have to kind of, what you can visualize, and this really is from this figure from their original Grunfeld paper, is look at the kind of, so if we have one eye in each of these kind of columns here. Okay, so they've actually sort of just extrapolated it up so you can kind of see. Look where the pinwheels, those columns of singularities, kind of fall. I think I remember it was like roughly like 80% of these pinwheels lay perfectly down the midline of each of these orientation columns. So it's actually very, very systematically organized in your visual cortex. But yeah, so that's the concept of the CO blobs and the whole pinwheel thing. It's just, when I was teaching that, it was a matter of sort of linking, oh hey, it turns out that these orientation columns come together in this pattern where we suddenly find these occasional pinwheel points, and these occasional pinwheel points correspond to, coincidentally, columns of neurons, these CO blob neurons that respond to color. All right, so I guess that was the last point here. All right, so hopefully that's helpful. That kind of took longer than I thought. All right, see you in class next week.